In this video, I'm going to give a basic introduction into the biotech industry, and this is going to be part one of a two-part series. And in this particular part, I'm going to give a basic overview of the different industries within the biotech industry and how they work and the challenges behind them. There are three major biotech industries. The first is the one that people most familiar with and associate with biotech is the pharmaceutical industry, but people tend to forget that biotech actually has a huge impact in the agricultural space. And more recently, we actually are seeing biotech have huge impacts in the industrial space. And so the first one I'm going to go ahead and do deep dive in is the pharmaceutical industry. And so within the pharmaceutical biotech space, it can actually be divided into multiple subsections. The first is the one that people are most familiar with. It's just a small molecule. There are antibodies, there are gene therapies, cell therapies, and vaccines. And so there are a lot more than this. For example, there are things like called recombinant proteins, but these are the ones I'm going to highlight today. And so the first one I'm going to do a deep dive into is a small molecule. And so oftentimes these are going to be the powders that you normally find at the pharmacy or the pills that you find at the pharmacy that people are most familiar with. These pills will have some sort of synthetically made compound that you can introduce into some sort of sick patient. And the idea here is that inside the patient, you have some sort of target cell that is inducing the issue within the patient. And the idea here is that these cells can have some sort of a surface receptor that these synthetic compounds, once they are within the patient, can interact with these surface receptors in a way that activates a pathway or deactivates a pathway in a way that induces some sort of therapy response or cures the patient of the particular issue that they're having. Another way that these synthetic compounds can work is you can have some sort of intracellular component. Oftentimes, is going to be some sort of enzyme. And the idea here is that it can have some sort of synthetic compound that starts interacting with this intracellular component such that you can have some sort of beneficial response within the patient. So some of the challenges here, one is the high failure rate of developing these drugs, and this is mainly because it's highly unpredictable to predict how these molecules are going to actually end up behaving within the patient during clinical trials. The second is identifying novel targets, and this is mainly because this industry has been along for such a long period of time that it can be really hard to identify new targets to go after. And the third is that it can also be very hard to get specific targeting at a particular location and limit the side effects. And so oftentimes these small molecules will interact with your target cell, but also interact with other cells within the body as well, and induce some undesired effects. And so because of this non-specificity of small molecules, this kind of drove the adoption of antibody therapies. And instead of having a synthetically made compound, these drugs are actually made of proteins called antibodies. And so they are these Y-shaped proteins that have these binding regions at the ends of them. And the idea here is that when you introduce them into a sick patient, and you have kind of the similar setup, instead of having a synthetic compound that kind of targets a cell and goes elsewhere in the body, you can have these antibodies that specifically only bind to the surface receptor that you're looking at, and it can activate a pathway or deactivate a pathway like you did similar to before. And now the interesting thing with antibody therapies is that they can introduce new ways of generating a therapeutic response. And so, for example, you can have an immune cell that would normally recognize the target cell that can be blocked by the antibody. You can also have some sort of cargo that can be bound to the antibody. And so instead of having a small molecule that goes everywhere in the body, it can actually specifically locate on the surface of the cell. And more recently, we've actually seen the popularity of something called bispecific. And the idea here is that instead of having these Y tips only bind to one particular target, you can actually have each end of the antibody bind to two different regions. And so if you introduce this into a sick patient, you can have a situation like, let's say an antibody binds to a cancer cell via one part of the binding region, and then you can have an immune cell that binds to the antibody on the other part of the binding region, such that an immune cell can actually start directly interacting with a cancer cell and start killing it. And so the challenges within the antibody space is the low-hanging fruit had kind of already been picked. And so at this point, the antibody industry has been very mature. We know how to, again, discover antibodies, manufacture antibodies, and know how to get them to market. What's actually just finding particular disease targets that have actually beneficial therapeutic properties is the main challenge right now. And in particular, a large part of the industry is looking for ways to have antibodies bind to something called GPCRs, which have huge therapeutic potential. And the idea behind GPCRs is that they are these surface proteins that are embedded within the plasma membrane. And the reason why they are hard to get antibodies to bind to is that oftentimes they have these very small accessible binding sites versus other surface proteins that have large surface components that antibodies can readily bind to. And so if we can find a way to actually target these smaller regions within the GPCRs, we can have a huge therapeutic potential here. And so the third main industry I'm going to do a deep dive in is the gene therapy space. And the idea behind gene therapies is that you're going to have some sort of virus that carries some sort of gene editing cargo. This can be, for example, CRISPR editing technology that you can introduce into a sick patient. And the idea here is that this virus can interact with some sort of target cell in a way such that the gene editing cargo gets introduced to the cell and then starts editing the actual genome of the cell. And so you can do things like, for example, edit sickle cells to cure sickle cell disease or cure Alzheimer's or things along those lines, right? And so there's a huge potential here within the gene therapy space. So some of the challenges here is cost of manufacturing these viruses, because oftentimes a batch can cost, or a particular treatment can cost like $1 million to actually manufacture. And so we need to find ways to bring those costs down. The second is actually having these viruses target a specific location. So right now we're kind of limited to having these viruses circulate within the bloodstream or go to the liver or something like that. But getting it to go into other regions and specifically target different regions of the body is a major challenge within the gene therapy space. The third is that these viruses are immunogenic, and so people are trying to find ways in order to introduce these gene editing cargo without the use of viruses. And the fourth is that these gene editing tools can't make larger gene edits. And so right now we're kind of limited to point mutations and smaller gene edits. But if we can get to a point where we can actually do full gene replacement, that can provide a huge therapeutic potential. Now, the fourth that I'm going to go over is the cell therapy space. The idea here is that if you have a sick patient, you can actually extract stem or immune cells from that patient. You can genetically modify or genetically engineer those cells in a way that if you're re-injected into the patient, they have some sort of therapeutic response. For example, if you had a genetically engineered immune cell, oftentimes this can be something like a T cell. These T cells can start recognizing the surface protein of, for example, a cancer cell and start killing it. If you're looking at the regenerative space and you have some sort of damaged tissue, you can take an engineered stem cell, have it engrafted within that damaged tissue. You can have them, grow up and start regenerating that tissue. For example, if part of your retina within your eye has some sort of damage that causes you to lose eyesight, you can actually have these regenerative medicines regenerate those particular regions of your retina and cure blindness within that patient. Another way that these stem cells can work is that they can actually start secreting something that actually causes other cells in the area to start growing or behave in a way such that it replenishes or repairs the damaged tissue. And so some of the challenges within this industry, one is the complexity and cost of executing the treatment, mainly because you're not having a system where you're manufacturing these upstream and you're actually going in with each individual patient having a personalized treatment that can be really complex and very costly. The second is that because you're creating a personalized treatment, it's really hard to create consistency from batch to batch. The third is actually keeping the cells alive during the actual treatment, mainly because these cells are very fragile, and you're not going to have too much time to genetically modify and reintroduce them before they start dying off. And so that makes it really challenging to actually execute these treatments. And because of these issues, you actually have huge regulatory barriers. mainly because it's really hard for regulators to recognize which ones are actually safe to pursue, and which actually have the kind of benefits that we were looking at. The fifth one is the one that became popular in COVID, and that's kind of the vaccine industry within the pharmaceutical biotech space. And the idea here is that you have some sort of attenuated or inactivated infectious disease. Maybe it's just some sort of virus or something that causes some other sort of disease. And the idea here is that you can introduce this into a patient. This patient develops some sort of immune response, and such that you don't get as sick if you get reintroduced to that virus natively when you get exposed to them when just like walking around during a normal day. And so some of the challenges with some of these more traditional vaccines is one is that it takes a lot of time to develop and manufacture these vaccines, and also the efficacy can be actually be very low. And so oftentimes you need to take multiple doses of the vaccine before you actually get the immune response that you want. And so this actually led to the development of mRNA vaccines, which became very popular during COVID. The idea here is that you can have these lipid nanoparticles that carry something called mRNA. And then the idea here is that these mRNA are much faster to develop and synthesize than these previously traditional vaccine modalities. And the idea here is that you can take these lipid nanoparticles that carry these mRNAs, introduce them into a patient. These lipid nanoparticles will interact with cells in the body, introduce these mRNAs. Because the cell has machinery to convert these mRNAs into attenuated versions of the disease target, these proteins can get released into the external environment or displayed on the surface of cells, such that immune cells can start recognizing it, and you can start developing an immune response and develop an adaptive immunity to the disease that you're looking at. So some of the challenges within the vaccine space, one is that while it's still a little bit better than the traditional vaccines that were previously made, the mRNA vaccines are actually still very complex to manufacture. Also, the stability of mRNA during storage is really challenging. So they tend to degrade very rapidly at room temperature. And so finding ways to have them within a chilled environment consistently, such that they remain stable over time, is going to be a huge challenge within this particular industry. And the third is that because this is such a new area of interest, we still have a very limited understanding of the mechanism of action and the potential side effects. And so we need to do more research into this. And so if you kind of look back at the major biotech industries, we're going to go ahead and move away from pharmaceuticals and then into the agricultural biotech space. And the agricultural biotech space can be subdivided into 4 different sectors. The first is fertilizers, the second is biopesticides, the third is fermented products, and the fourth is cultured meats. And the first one I'm going to go over is the fertilizer industry. And the idea here is that if you are growing some sort of crops on some sort of farm, you need nutrients in the soil for the crops to grow properly. And so what you can do is if your soil doesn't have enough nutrients, you can take some sort of organic fertilizer that's composed of some sort of manure or compost. You can reintroduce that into the soil. And the idea here is that this compost contains extra nutrients that allow the crops to grow more effectively than if they had not had this fertilizer within the soil. So the issue with some of these organic fertilizers is that there are still oftentimes not enough nutrients, and the amounts of nutrients can vary from batch to batch. Because usually you're taking this from something like compost, right? And it's going to be hard to control how much nutrients are going to be there from batch to batch. And so in order to make sure that there are enough nutrients and more consistently enough nutrients, a lot of companies started making these inorganic fertilizers that are synthetically made, that you can introduce into the soil, that ensures that there are enough nutrients for the crops to grow and have much more consistency from batch to batch. Now, the issue with a lot of these synthetic fertilizers is that oftentimes they can introduce too much nutrients and introduce runoff and actually start damaging the environment. And so we need to find a way to actually make sure that only the right amount of nutrients are available to the crops at the right time. And so with a lot of these gene editing technologies that have been developed over the past decade or so, we've been able to actually engineer microbes that can behave as these fertilizers. And the idea here is that you can make previously inaccessible nutrients available to plants right? And so you can do things like take atmospheric CO2 and fixate it such that they're readily available to the plants. You can take insoluble phosphorus and make them soluble, such that they're all more readily available to plants. And the idea here is that, unlike with synthetic fertilizers, you don't dramatically change the environment. You're simply just taking the nutrients that are actually available and actually present within the environment and just simply making them accessible to the plant, rather than reintroducing a completely new nutrient composition. And so another cool thing is that these nutrients can actually be made available to the crops much more dynamically. And so it can, over time change the amount that's actually converting from a non-available format to an available format to the plant. And so the idea here is that only a right amount of nutrients are available at the right time. And so this is going to be like much more efficient than what was previously used. And so some of the challenges within the fertilizer space is one that it's really hard to ensure that the microbes are actually alive by the time the fertilizer is used. And because, usually you're going to be manufacturing things and you need to transport them to the end user, oftentimes going to be farmer. You need to find a way to make sure that those microbes are actually alive when they are actually used by the farmer. The second is that it's really hard to develop any sort of assay, reaction, or test to actually measure what the microbe is actually doing. And so finding ways to actually measure the fixation of nitrogen, for example, or the conversion of insoluble phosphorus to soluble phosphorus, that's not actually quite straightforward as it may seem. And the third is actually trying to find ways to convince farmers to adopt new methods and practices. And because they're oftentimes very resistant to new technologies because it's such a huge risk if it ends up going wrong. And so you need to have like very extensive data and have a lot of patience in convincing these farmers in order to adopt some of these newer practices. And so another area of interest within the ag bio space is biopesticides. And so the problem when you're starting to grow up crops is oftentimes you have these bugs that if they are exposed to the crops that you're looking at, oftentimes they can start eating away at the crops and actually start to cause them to die prematurely before harvest. And so this is going to cause a lot of inefficiencies with our ability to grow these crops and harvest. And so, what a lot of companies did in the past was that they were able to make these microbes or take these synthetic compounds that, if the bugs are exposed to them, it can either make them unhappy and go away from the plant or kill the bugs altogether. And so, the idea is if you can, spray these synthetic compounds onto the crops, these crops can remain viable for longer periods of time. And so the issue here is that a lot of these previously used biopesticides were very nonspecific. And so they would not only go after the bug that you're looking at, but would go after bugs that that you're not looking at that are actually beneficial to the crops. And so it's really hard to get a lot more specific targeting towards the bug that you're looking at. And so more recently, what we've been able to do is actually engineer microbes that can go after the bug that you want to kill. But one that I actually want to take a little bit deeper dive into are these nanoparticles that have like double-stranded RNA in them. And the idea here is that you have these double-stranded RNAs that are specific and only interact with the bug that you're looking at versus other bugs. And so the idea here is that if you have a bug that ingests these nanoparticles, these double-stranded RNA can actually start disrupting proteins that are specific to that bug via a mechanism called RNAi. And so if a different bug were to ingest the same nanoparticles, it wouldn't affect them, but would affect the bug that you're looking at. And so this is going to make things a lot more specific and reduce the amount of environmental damage that you induce to the crops. And so some of the challenges here is one is it can be a little bit challenging to ensure that the nanoparticles are actually ingested by the bugs. And so making sure that they're in the proper location, for example. And the second is actually the microbes can actually start adapting resistance to these particular these double-stranded RNAs, and so you need to design them in a way such that they don't become and evolve resistance to the double-stranded RNA over time. And so a third and up-and-coming industry within the biotech space is the fermented products. And the major motivation here is that our making our food is like very time and resources sensitive. And so oftentimes we extract a lot from the environment, but we don't have any really good ways of replacing what we took. And so the idea here is that if we can just take the raw components of any food and just like engineer them or combine them together to make the food that I'm looking at, you can actually make fully synthetic food. And the idea here is that we can make food much more quickly and efficiency. And so instead of simply just relying on nature and growing these crops outside, we can actually start synthesizing a lot of the food that we like within a laboratory setting or within a factory floor. And so the idea here is that we can take like a microbe, right? We can actually have these microbes start making these raw components. And so the idea here is that if you have atmospheric CO2, you can have this atmospheric CO2 go into sugarcane. This sugarcane can be made into sugar that can be fed into an engineered microbe. These microbes can be grown up in these large fermenters, and then you can have these microbes produce these raw components at large volumes, and then be used to make these synthetic food products. And so the idea here is that you can create a much more sustainable infrastructure over longer periods of time. And so what are some examples of these fermented products? Well, one that people are looking at is, for example, engineering these proteins that are important to the stringiness or the texture of cheese. Or you can have like microbes that make fats that are important to the flavoring of chocolate. You can actually have companies that make these certain sugars that can later be made into honey. So you can have like fermented honey, for example. And so there's a lot of really interesting things that you can actually do with these fermented products. And so some of the challenges here, one is that it's really challenging to make things taste like the real thing. And so an end user will not actually adopt if they don't like the taste or if it doesn't taste like the more natural version. And so that's going to be the major challenge within the fermented product space. The second is the cost and time to actually develop these things. And so such a huge undertaking that you need to make sure that the end user is actually going to actually adopt by the time you actually make these fermented products. And the third is actually you need to find ways to have alternatives to sugarcane, and mainly because it's a very inefficient way of introducing sugar as an energy source to the cells, and also very time intensive and resource intensive. And so if we can find ways to actually just directly take atmospheric CO2 and have them as the carbon source to the cells, this can be really beneficial for this particular industry. And the fourth one I'm going to go into is the culture meat industry or lab-grown meat industry. The idea here is that you have some sort of animal that you extract some stem cells from. You can differentiate these stem cells into a particular meat cell, and so maybe this is like a muscle cell that's for beef, for example. Uh, then you can grow them up into these large tanks or cell culture environments. And then you can, make enough and such that you can have certain scaffolding and making them into like a fully edible pieces of meat. And so some of the cool things you can actually start doing with this, other than making a more sustainable way of making meats, is you can do things like, okay, you can start making more unique lean meat to fat ratios that were not possible before. You can do things like enhance the nutrient compositions. You can also introduce new flavorings, and so you can actually make meat naturally garlicky or spicy, or things along those lines. And so there's some really cool things that can start doing once we start thinking about the future of the meat industry. You can also do things like meat fusions, and so you can like combine a beef with a chicken or turkey with a duck, and so you can start making these like unique meat products that weren't possible before. And so some of the challenges within this industry, the big one is going to be the cost of media equipment. And so these culture meats, even though we can like make them, they're just simply too expensive for the end user to actually adopt. And so we need to find a way of actually reducing the cost of media. And then it's mainly because the media and equipment that we use are pharmaceutical grade and actually not designed for this purpose. And so if we can get away from pharmaceutical grade media and equipment, we can actually make these things much more cheaper and much more economically feasible. And the second is regulatory. And because it's such a new industry, there's a little bit of unclarity within the regulatory environment at the moment. And so we need to find ways of making sure that we have a much more clear guidance on the regulatory components. And the third is avoiding the GMO label. And so while we can do like these amazing things with the culture meats, if we start over engineering the cells, they can get a GMO label. And oftentimes there's a negative public connotation that comes with that. And so we need to find ways to engineer these cells in a way such that they don't have the GMO label, so that they can be readily adopted, while also introducing these unique properties that we want to have. And so that's going to wrap it up for the agricultural space. And so the third and final one that I'm going to go over is the industrial biotech space, which is the one that's growing the most and is the one that people are actually not looking into enough. And so this is going to be broken down into several different sectors. The first is going to be enzyme. The second is going to be small molecule. The third is large molecule. And the fourth, I'm actually going to coin this one called cell machine. And so looking at the enzyme space, the basic idea behind enzymes is that you can have some sort of substrate that interacts with an enzyme, changes the properties of the substrate, and makes some sort of product, right? And so you get a substrate that interacts with the enzyme such that it changes the properties and becomes something else. And so if you, for example, you can take some sort of synthetic or small molecule compound, it can change the structure of that compound. You can have some sort of polymer that's broken into pieces, or two different monomers that are bound together to form a polymer. And so that's the idea behind these various enzymes. And so what you can do is you can have genetic material encoding for an enzyme. You can introduce that into a microbe, just that microbe can start producing those enzymes that you want, and then you can use them for various different applications. And so some of the traditional enzyme industries, first is like the food and beverage industry. For example, when we used to make milk, we used to have to go to a cow and extract the enzymes from a cow, and kill the cow and have these inefficient processes to use our enzymes for the actual processing of milk. But now we can actually just engineer microbes that can even have better enzymes where we don't need to kill cows, and is much more sustainable over the long term. So it's much more beneficial to actually have these microbes engineer these enzymes rather than extract it from animals or the environment. Another one is that they can be used to synthesize pharmaceuticals. Oftentimes they can be in detergents. And so back in the day, we actually use like these fats that you also have to extract from animals. And now we can actually just engineer enzymes that have much more properties better properties that we can introduce into detergents. We can also use enzymes to make textiles for clothing and fabrics and things along those lines. And it can actually be important for things like paper production. And so more recently, we've actually seen some more exciting environmental applications of enzymes. For example, you can actually take enzymes that are naturally found in plants that convert CO2 to glucose for plants, and try to mass produce that. And so we can actually have enzymes that do carbon capture. You can do things like have enzymes that degrade plastics. You can have things like isolate lithium, because getting enough lithium to make enough batteries for the sustainable economy long term is going to be a huge challenge. You can have enzymes that clean up water. You can also have things like enzymes that take out air pollutants. And so there's a lot of really exciting applications here for the enzymes. And so some of the challenges within this particular industry is one is making enough enzyme, and mainly because unlike the pharmaceutical space where you can charge a lot for a small amount of product, enzymes are not as monetarily valuable. So you need to make enough at a cheap enough cost, such that it can actually be economically valuable. And that's actually going to be a challenge. And I'll talk about that in part 2 of this particular series. And so people are actually trying to find ways to recycle these enzymes. And so that will reduce the amount of enzyme that we actually have to make. But also, like, designing and screening enzymes is also very hard and challenging. And so because like proteins are very complex, it can be a little bit hard to actually design an enzyme that has the actual catalytic response that you want to look at. And so more recently, we've seen the development of the small molecule industry. And so the motivation behind this is that, what if like one enzymatic reaction isn't enough? What if you need multiple enzymatic reactions to get the end product that you're looking at? And so what you can do is you can actually engineer a series of enzymes that takes the substrate and then makes multiple edits to it to give you the product that you want. And this is going to be a process called pathway engineering. And the idea here is that you can have a microbe hold all the different enzymes within it and have this whole pathway within that microbe such that the microbe itself can, when exposed to the substrate, can convert it to the product that you're looking at. And so some of the different applications you can do with this, one is like natural medicines. And so, for example, you can make, instead of going to plants to make cannabinoids and psychedelics and things along those lines, you can actually have microbes that synthetically make them. You can have microbes that make biofuels. You can make like flavorings and nutrients. And so one thing people tend to forget is that microbes oftentimes are used for vitamins and supplements and things along those lines. Another thing you can do is you can have like microbes make unique fragrances. You can actually have microbes even make metals, and so you can have make like iron oxide or potentially even like something like gold in the future. And so that provides some exciting applications. And so, but like, why would we want to do this? And so why don't we just use the traditional ways of actually making these different molecules? And so one motivation here is that you can create, again, a sustainable bioeconomy. And so you have a way of sucking up atmospheric CO2 from the environment. But another thing to consider is that you can actually start having substrates that are much more sustainable or in abundant amounts. And so you, instead of using, traditional processes where you're using some of these inefficient or, very rare inputs, you can actually engineer enzymes that can have unique pathways such they have much more sustainable inputs. And so, and then the second kind of motivation here is that if you look at like kind of traditional chemical processes, they have like very limited strategies to make novel or new chemicals. And so what cells provide are new precise ways of actually editing these materials. And so even though right now we are focused on engineering microbes to make chemicals that are available today, we can actually start thinking long term about making chemicals that weren't possible before. And so that's going to be highlighted actually more in something like the large molecule section, where instead of taking a substrate and make small modifications to that, or to make a small molecule end product, you can do something along the lines of binding multiple subunits together to make these larger polymer matrices and make what's called like a large molecule. And so some of the applications here, right, is that you can take a bunch of like monomers, have them introduced into the cell, and then make these larger structures. And then some of the industries that are being popularized by this, one is like the biodegradable plastics. Another one is you can have like mucus-inspired lubricants that have unique properties. You can make electrical components, so you can actually think about making like degradable batteries, for example. You can make new composites, and so you can like mass produce spider silk or things along those lines, or chitin and other different materials that have unique properties that you can use for different applications. And you can also do things like making cosmetics, and so that's actually a really popular space within the large molecule industry. And so the cell machine is actually something I'm going to coin at the moment. And the idea here is if you take a pathway that can make a large molecule or a small molecule and put them into a microbe, instead of having the cell produce the end product, you can have the cell be the product itself. And so you can do things like make fertilizers, as we kind of already discussed before, or you can do things like a bioremediation. And so if you have some sort of oil spill out in the ocean, you can have microbes that can go ahead and actually clean up those oil spills. And there's like another cool one that's up and coming is you can make something like self-healing bricks. And so if you have some sort of brick that has cracks, you can have these microbes embedded within these bricks that can actually start repairing these bricks over time. And so these things are much more long-term and a lot more visionary, but it's good to keep in mind that these are some things that people are actually looking at within the industrial biotech space. And so some of the challenges with a lot of these pathway applications is one is solving actual market needs. And so right now we're just kind of, again, synthesizing products that are already available in the market. And so making sure that we actually are using the systems to actually make products that the market actually wants and actually needs is a huge challenge right now. The second is the time to market, and that's because we're actually competing against more traditional processes that are much more fast to make end products. It takes them a lot more time to actually engineer microbes to make these products, and so we need to find a way to reduce the time to market. The third is the lack of genetic tooling in order to make the products that we want to look at. And then the fourth and the more important one is the cost to develop and manufacture these. And so even if we could make these products, that solve actual market need, oftentimes they're a little bit too expensive for the market to actually adopt. And so we need to find a way to reduce those costs. And so that's going to wrap it up for the overview of the different sections. And so, make sure you comment if you think anything was missed, and also be on the lookout for part 2. We'll actually go overview how these products are made and the different challenges behind that. And so make sure you go to the Carl Lab UCLA channel on YouTube. And also I'm going to be posting this on my LinkedIn as well. So again, please be on the lookout for part 2, and I look forward to seeing you guys there.